Welcome to class 11 students. This is the continuation part of the chapter chemical constituents of living cell. In my previous video I started with the topic of enzyme, and I have discussed what enzymes are and how what are the components of the enzyme, which are apoenzyme, which is the protein part, and coenzyme, which is the non-protein part. Both of them make the total enzyme, which is known as holoenzyme. Then I have also discussed about the allosteric enzyme, what is allosteric site, allosterism. I hope everything you have understood and all these things are clear to you. Today, the next topic that I am going to start from this topic enzyme, under this topic enzyme, is the classification of enzyme. That part is very important, but before going into the classification of enzyme, let me tell you a few small things that you have to learn from your book. Is firstly, obviously you are going to learn the definition of activation energy. Then you will have to learn the definition of enzyme and the chemical nature of enzyme, whatever I have discussed. Then the definition of cofactor and prosthetic group, allosteric enzyme definition, all these things I have discussed, and I want you to learn it from your book. Next, you will find the difference between apoenzyme and coenzyme, which is very important. That also you will have to learn. And next we will talk about the difference between inducer and inhibitor. So I have already said that these are the effector molecules. Any molecule that brings about a change within an enzyme is known as effector molecule. And there are two types of effector molecule: inducer, which induces or stimulates the activity of the enzyme, and another one is the inhibitor, which inhibits or stops the activity of the enzyme. Okay, so the difference between inducer and inhibitor, that also you will have to learn. And a few other terms like proenzyme, anti-enzyme, isoenzyme, all these things I have already discussed in the online class. Proenzyme is basically the inactive enzyme from which enzyme is formed. Anti-enzyme are those agents which work in an opposite manner to the enzyme. For example, they try to destroy or inhibit the activity of the enzyme. And then you have the isoenzymes. Another topic that you have is the different types of digestive enzymes. This is very easy. I have already discussed this in online class. The three different types of digestive enzymes are proteolytic enzyme, lipolytic enzyme, and amylolytic enzyme. From the name only you can understand what they digest, what these enzymes digest. And the proteolytic enzymes digest protein, lipolytic enzymes digest lipid, and amylolytic enzymes digest carbohydrate. And you will have to learn the definition, one line definition of each, and one or two examples of each. Okay, and now we can move on to the next topic, which is the classification of the enzymes along with their example. Okay, so let's start. Types of enzymes with their examples. So as you can see, these are the types of enzymes. This enzyme class is the types of enzymes. So the first one is hydrolase. This is the first one, hydrolase. So what does it do, or how does it react? According to the definition, enzymes which bring hydrolysis of variety of compounds by the addition of water molecule are known as hydrolases or hydrolyzing enzymes. So here you can see it is written that hydrolysis. So what is the meaning of hydrolysis? Hydro means water and lysis means breakdown. So when there is any breakdown involved due to the addition of water molecules, Catabolic, as you know, the meaning of catabolic is breakdown. So the breakdown of any substance due to the addition of water molecules is known as hydrolysis, and this is done by enzymes which are known as hydrolase. Okay, before going further into this topic, I will like to tell you one thing, that any enzyme, commonly all the enzymes end with the suffix ase, as you can see, AAC. Okay, in example also you will see that protease, lipase, this one, all have ases at their end. Okay, but there may be some exceptions where the name of the enzyme does not end with ase, but commonly it does so. And then even if you don't know the name of an enzyme, just by if the ase is there, you will get to know that this is an enzyme. Okay, so example of hydrolysis is lipase and protease. Clear? Next one, moving to the next category, is isomerase. Number 2 is isomerase. So what are isomerase? How do they work? These are enzymes which catalyzes the isomerization reaction. What do they do? Here it is written that rearrangement of atoms within a molecule. So isomerization is that only, rearrangement of molecules within a rearrangement of atoms within a molecule. So that action, that reaction is catalyzed by whom? By isomerase enzymes. And examples include phosphoexoisomerase. Okay, next one, lyase. Next one, number 3 is lyase. These are the enzymes which catalyze the addition or removal of groups to or from substrates containing the double bonds by the process other than hydrolysis. So this is more or less like hydrolase, but it does not work just like hydrolysis. It splits chemicals into smaller parts without using water. So as I said, it is not, it splits chemicals into smaller parts, but not exactly hydrolysis, where breakdown is done by the by the addition of water molecules. This is also catabolic in nature, but without using water. Clear? So examples include decarboxylase and aldolases. Moving on to the third one, that is fourth one, which is oxidoreductase. These enzymes catalyze oxidation and reduction reaction in the cell. So oxidation reduction reactions, also together known as redox reaction, they help in the transfer of electrons or hydrogen atoms from one molecule to another. Okay, and examples include dehydrogenase, oxidases, etc. Fifth one, that is synthetase. The other name of synthetase is ligase. Okay, this is the other name. This is the more common name, more common than synthetase. So ligase is the other name of synthetase, and these enzymes catalyze the linking together of two molecules by splitting a phosphate bond. Okay, so here also it is written that joining of two molecules by the formation of new bond. Joining of two molecules take place by formation of new bond, and anabolic means to make up, to create something, which is just the opposite of catabolic. Okay, catabolic means to break down and anabolic means to create something. Clear? And the examples include DNA ligase, DNA polymerase. These are all required during the process of DNA replication, and they all act as ligase enzymes which join two molecules together. Okay, last one and the sixth one is transferase. From the name only you can understand these enzymes catalyze the transfer of any particular group, functional group, from one molecule to the other molecule. Okay, moving a functional group from one molecule to the other, just what I said. Okay, and examples include kinases, transaminase. So all you have to do is you will have to mainly learn these two portions, this enzyme class and reactions catalyzed. It will be better if you can remember the examples, at least one. And these are DNA ligase and DNA polymerase. These are most important than any of these. So if you can learn this chart, if you can write down this chart in your copy from the video, then it will be very good. I don't know if your book has any chart or not similar to this. If it has, so you can do it from your book as well. Okay.